will attach to this chromosome so that fibers from one pole attach to one of the sister chromatids. Fibers from the opposite pole attach to the other sister chromatid. All right? So our chromosomes become attached to the kinetochore fibers. I'm going to draw in all 4 of our chromosomes with those attachments. Remember, I've got two long chromosomes, 2 short ones. I have to draw them in a minute. And then I'm going to connect, I'm going to connect my kinetochore fibers properly. I want to make sure they're connected properly. In fact, a cell double checks that its kinetochore fibers are connected properly. Otherwise, it could be a real problem later on. All right, let's connect our kinetochore fibers here so that they're properly connected. This guy needs one, this guy needs one here. Okay. In addition to those kinetochore fibers that are connecting to the chromosomes, we have non-kinetochore fibers. The non-kinetochore fibers are fibers that extend from the poles toward the center of the cell, but they're not attached to anything. They're going to be useful. They overlap even at the center. Okay, so there's an area of overlap, but they're going to be useful, but they're not connected to the chromosomes. Now I've got kind of a big fat mess for my prometaphase. What happens next? All right, let's move into metaphase. So let's redraw another cell and think about what's going on during metaphase. I'm throwing my markers on the floor because I don't have a nice chalk ledge in my basement whiteboard, so I need to get that. During metaphase, there's kind of a tug-of-war going on, and eventually the chromosomes become aligned at the midline of the cell. So each chromosome, remember, is attached to fibers from opposite poles, and these kinetochore fibers kind of pull the chromosome back and forth a little bit, but eventually all of the chromosomes end up so that they're aligned with their centromere in one plane of the cell. We call that plate the metaphase plate. So I'm going to write down that the chromosomes align at the metaphase plate because of this kind of tug-of-war action that's going on by the spindle fibers. All right, let me draw in my chromosomes a second. So I end up, the legs of the arms of the chromosomes don't necessarily look exactly like they're all lined up perfectly, but I'm just going to draw them in. I had one long green chromosome, one short green chromosome, one long red chromosome, one short red chromosome, right? In my cell. I have 4 chromosomes at this point. And I have to draw in my kinetochore fibers that are properly attached to these chromosomes, right? And I've got my centrosome at each pole. And I want to draw in some non-kinetochore fibers. They're going to be important in the next phase. Okay? Duh, that's metaphase. What's going to happen to these chromosomes? Any ideas? Okay, we've got a chromosome made up of 2 identical copies. And what's going to happen to them, do you think, in anaphase? These identical copies are going to separate and move toward opposite sides of the cell, opposite cells, really, right? That's what we were expecting, and that's exactly what happens in anaphase. Okay, in anaphase, we want to draw our cell a little bit differently. We want to draw our cell kind of long and skinny. This is an animal cell that we're following because the cell starts to stretch out, so we're going to have to record that. And we want to draw our chromosomes differently. At this point in anaphase, the sister chromatids separate from each other, and sister chromatids move toward opposite sides of the pole. Sister chromatids separate and move toward opposite sides of the pole. Remember that there's a protein called cohesion that's kind of holding the chromatids together. That protein is destroyed. There's an enzyme that breaks it down, and we see the kind of separation of let's see, separation of each chromosome or each chromatid, and they kind of look like little V's as they're being pulled toward the opposite side or the opposite pole. So I've got a short green, a long green and a short green chromosome. I've got a long red chromosome and a short red chromosome, right? And we need to write this down, but I'm kind of doing the drawing first. Now, what's going on here? I'm going to draw that out. And I'm going to write sister chromatids separate from each other because that cohesion protein is broken down by an enzyme. Sister chromatids separate and move toward opposite sides of the, toward opposite poles. We'll just write opposite poles of the cell, all right? How are they moving toward opposite poles of the cell? Well, there's some different ways that they can move, but they depend upon these microtubules. These microtubules shorten, and the chromosomes then move toward opposite poles. So the microtubules can either shorten on this end, and they kind of get reeled in, if you will, or they can shorten on this end, and as they get shorter, they kind of get, it's kind of like Pac-Man. The chromosome is kind of walking toward this pole. So they move this way, all right? All of these are they called chromatids or chromosomes at this point? We have one chromosome made up of two chromatids, like we do in metaphase. As soon as these chromatids separate from each other, each chromatid is now like a mature chromosome. So each chromatid becomes a chromosome, and one chromosome is moving in one direction, the other chromosome is moving in the other direction. So at this point, if we really want to be technical about it, there are 1, 2, 3, 4, 5, 6, 7, 8 chromosomes in the cell. There won't be for long. We're going to separate these two parts of the cell into two. But at this point, this thing is called a chromosome because it's separated from its sister. Bye, sister chromatid. We won't see you again. So the sister chromatids separate and move toward opposite poles. The second thing that we want to write down is that the cell elongates due to the action of the non-kinetochore fibers. So I wrote down in parentheses here, due to the action of the non-kinetochore fibers. What happens here is these non-kinetochore fibers are extending toward the poles and they're overlapping. What happens with them is they ratchet by each other. This takes a lot of ATP. They ratchet by each other and they stretch out the cell as they push ratchet by each other or push out away from each other. And they're extending or stretching the cell, so it makes it easier for this cell to separate, and it makes sure that the chromosomes end up in the right places, right? We want to have 4 chromosomes in this side of the cell and 4 chromosomes in that side, and we better have a long green one, a short green one, a long red one, a short red one on each side. Otherwise, we got problems. We're almost done. All we have to do is complete the process with telophase and cytokinesis. I'm going to erase, and we're going to draw telophase and cytokinesis so that we can finish mitosis and see how one cell divides into 2 cells with identical genetic material. So we're following this cell that has 4 chromosomes in it. I'm going to draw telophase like this. Okay, we're coming from anaphase, right? In telophase, a whole bunch of things happen. One of the things that happens is the nuclear envelopes, all those little pieces that were separated back in pro-metaphase, these nuclear envelopes start to reform. So I'm going to write that down. The nuclear envelopes reform. It's not like some sort of religion, but they just come back together again. around the chromosomes, we hope. Okay? The nucleoli reappear. The mitotic spindle disappears. So no more spindle. I'm just going to draw the centrosome, but in each forming cell. But I'm not going to draw a spindle anymore. And the genetic material, the chromosomes, that are full-fledged, tightly coiled chromosomes, they start to uncoil. And they start to decondense, if you will, and eventually they'll become stringy masses called chromatin again. I don't really want to draw that yet because I want to see my chromosomes. But I'm going to write it down that the chromosomes uncoil to form chromatin once again. Chromatin, okay? The stringy mass of material. And I'm going to draw in my chromosomes so I can see what I have. From anaphase, I should have 4 chromosomes in each cell. My chromosomes now don't have a sister chromatid, so they just look like a stick with a centromere, okay? I should have a long green, a short green, a long red, and a short red in each of the cells. That would be good. Each cell then has 4 chromosomes, which will become the chromatin, all right? And 5. The last thing that happens is cytokinesis. Cytokinesis occurs, the separating of the cytoplasm, right? As this cell is going through telophase, a groove starts to form, and this is an animal cell. A pinching process starts to take place in animal cells, and that process is called cleavage. So we talk about a cleavage, cleave to cleaver to break, right? A cleavage groove forms, and essentially we've got some motor molecules that cause a constriction, kind of like you're squeezing a belt tight or something like that, right? And eventually that cleavage groove causes the 2 cells to be pinched apart, and we end up with 2 cells. They're about half the size of the original cell, but these 2 cells should be genetically identical. Each cell should have 4 strings of chromatin, or each cell has 4 chromosomes in it, right? We hope so, at least. And it has a centrosome, and it will have the nucleoli. So we have 2 genetically identical, identical cells. Yay! We did it. We went from 1 cell to 2 genetically identical cells. Mitosis is happening in cells that are just trying to make copies of themselves. Cells that are like skin cells, making more copies of themselves, right? If you're a plant and going through mitosis, plants have cell walls, and so they can't just kind of pinch their pinch their cytoplasms apart by cleavage. They have to have some other way of doing cytokinesis. They have what's called cell plate formation. What happens in cell plate formation, if you've got these 2 cells this 1 cell that is trying to separate, is that there are these little little vesicles that come from Golgi apparatus, actually, that move toward the midline of the cell and fuse together and become essentially the double membrane that connects to the rest of the plasma membrane of each cell to separate each cell completely apart from each other. Eventually a wall forms between them as well, but this is just the plasma membrane. Now we have 2 cells. So the process of cytokinesis in plants is called cell plate formation. We still end up with 2 genetically identical cells made from 1 cell. That's what mitosis is. I think we've covered the basics of mitosis in these videos, and we will wrap up the chapter 12 with a regular PowerPoint video. Thanks so much.